I would like to start my presentation with an issue by highlighting an issue that our generation faces, and that is climate change. And that is not working. And that is climate change. we all know that the situation is only getting worse, and one of the main reasons for that is the use of fossil-based materials. And while there is no planet B, there is a plan B, and that is the replacement of fossil-based materials with bio-based analogs. To this end, plant biomass, and more specifically wood, or Sweden's gold, is an important and promising alternative. One of wood's major components, lignin, which comprises up to 30% of its biomass, is a multi-ton byproduct of the pulp and paper industry. And currently, its main use is the production of energy for the mills through combustion. Although major efforts have been made during the past years for the valorization of these abundant biopolymers. However, one issue might be that the structure of lignin highly depends on the extraction method used. So after pulping processes, most of the times we end up with highly heterogeneous polymers that have intense color or smell, properties that further limit down the applications in which lignin can be used. So since structure-property relationship is important for material applications, we have the alternative to turn to lignin-first approaches. And in contrast to the current approaches, lignin-first approaches prioritize lignin extraction. And we can use them in order to target specific structures of lignin directly in the biomass. That way, we can reduce the amount of chemicals and energy consumption that most of the cases come together with post-extraction modifications in the current approaches, in which the biomass is first completely decomposed, and then we try to valorize the components. Several years ago, United Nations formulated these 17 Sustainable Development Goals, and this can help decelerate climate change, but more importantly, they can lead to the sustainable development of that can be implemented not only to the environment, but also the economy and society. In this thesis, we try to address as many of these goals as possible, and we think that the results can have direct and indirect positive impact in the goals that you see here. For example, by understanding fundamental aspects of lignin, we can promote lignin-first approaches, which can implement new technologies for lignin extraction, leading to them creating job opportunities in newly developed infrastructures. To start from the beginning, this is the most classical representation of the structure of wood. And here you can see the primary cell wall, which comprises polysaccharides, cellulose, and pectins mostly. And inside the primary cell wall, we see the 3 layers of the secondary cell wall forming. And the secondary cell wall also comprises polysaccharides, cellulose, and hemicelluloses, but it also contains lignin, which is a polymer of phenolic compounds. In the model here, we can also see the warty layer or lumen, and also the connecting area between 2 adjacent cells called the middle lamella. And the middle lamella contains, again, pectins and lignin. While this is a very informative model, it doesn't tell us much about the interactions between wood's major polymers. So moving to a more recent model, we can see that cellulose, hemicellulose, and lignin are in close contact in the cell wall, and they can interact through space and through the formation of covalent bonds. The interactions between lignin and polysaccharides or carbohydrates are called lignin-carbohydrate complexes. And here in this model, you can specifically see a covalent bond forming between lignin and the polysaccharide. The focus of this thesis is lignin, and its biosynthesis starts in the plant cell with an important amino acid, phenylalanine. Starting with the deamination of phenylalanine and successive additions of phenolic and methoxy groups on the aromatic ring, and finally with the reduction of the carboxylic group on the side chain of the amino acid, all of which are controlled by enzymes in the biosynthetic pathway, we end up with these 3 alcohols, paracoumaryl, coniferyl, and cinambyl. These alcohols only differ on the number of methoxy groups on the aromatic ring, and they comprise the three major lignin building blocks, or monolignals. From paracoumaryl alcohol, we have parahydroxylphenyl, or H units. From coniferyl alcohol, we have guacyl, or G units, and from synapyl alcohol, we have syringyl, or S units. And these are the three major monolignals that we find in the woody tissue. However, we can have other rarer monolignals as well. And these can come from the same or different biosynthetic pathways. These rarer monolignals can have simple structures, such as the C unit from cafe oil alcohol, or they can have more complicated structures, such as, for example, tristin. And these rarer monolignals are not usually found in the woody tissue, but rather in grasses or in the bark or seed coats, for example. The monolignals are polymerized in order to give the final structure of lignin, and this is a process initiated by oxidative enzymes, namely peroxidases and lacases. The role of the enzyme is to introduce a radical on the monolignal, which is then delocalized via resonant forms. When two radicals meet, they can couple, forming a covalent bond. Then we have some rearrangements, such as, for example, rearomatization, to end up with the lignin interunit linkages. With the mechanism here, you can see the formation of the most abundant lignin interunit linkage, which is the beta-O4. Lignin polymerization is thought to occur via an endwise way, meaning that a new radical is incorporated at the end of an already propagating chain. And following this or very similar mechanisms, we can have the formation of the other major interunit linkages. So we have the beta-O4, the benzodioxazine. We can also have carbon-carbon or condensed bonds, such as the beta-5 and beta-beta. And we can also have spirodinone and 4-5 structures. The structure and size of lignin have been a point of controversy for several years now, and started with a large and branched model suggested by the work of Adler in the 70s. We move on to more recent studies, for example, in 2011 by Professor Cristini, suggesting that lignin is rather small and linear. Then a bit later in 2019, the work of Ralph suggests that lignin again has a linear polymeric structure, only to return to a more branched model suggested by the work of suggested by Balaxon in 2022. It is important to point out here that most of these models come from characterization of extracted lignin, and the extraction has an impact on the structure of lignin. During these past 4 years, I had the opportunity to work with different projects and focus on different aspects of lignin. So today, starting with the first 2 projects, I will talk about lignin biosynthesis and interactions with the polysaccharide, then move on to the impact of extraction on the structure of lignin. And finally, I will try to showcase how lignin can be used in high-end applications. So the first 4 projects focus on understanding lignin structure and interactions from a fundamental research point of view. And then the last 3 projects highlight the uses of lightly modified lignin in applications. To start with lignin biosynthesis and interactions, we employed a Norway spruce tissue culture, the cells of which only produce a primary cell wall, but they have the ability to secrete monolignals that then polymerize in order to give extracellular lignin, or ECL for short. The experimental setup looks something like this. We have cells growing on solid medium, and then we transfer these cells into the liquid medium of the liquid medium to form a suspension culture. Then lignin is polymerized in the liquid medium of the suspension culture in order to give ECL. And its ECL is simply collected by centrifugation. With optical microscopy experiments with which we stained the monolignals purple with the Weissner test, we confirmed that the cells that were coming from the solid cultures did not produce any monolignals, but the cells collected from the suspension cultures did produce monolignals, here and here. One of the greatest advantages of this system is that there is no secondary cell wall formation, meaning that no lignin deposition occurs, which means that we don't need to extract lignin in order to characterize it. And not only that, we have the opportunity with this system to study the interactions between lignin and the polysaccharide during lignin polymerization. And we did that biomimicking the lignin polymerization environment with the addition of a secondary cell wall hemicellulose, namely xylem. In order to study the impact of xylem on lignin biosynthesis, we added the hemicellulose in the solid medium of this culture. And actually, we saw that the cells that were growing in the xylem environment were producing monolignals, even though these were not secreted in the solid cultures to produce ECL. Then we wanted to see the impact of xylem on lignin structure. So we added the hemicellulose into the liquid medium of the suspension culture. And we also created two different compositions of the liquid medium to test, again, the impact that would have on lignin biosynthesis and structure. In general, we tried several different treatments with different liquid medium compositions and xylem provision and different xylem concentrations. And a very small sample of these you can see in the table here. So our reference system is ECL collected from the liquid medium of suspension cultures that contained no xylem. And the Vita4 content was around 30%. And the molecular weight was rather small, around 1,900 grams per mole. Then we have the same medium composition, but then with the addition of xylem. And we saw that the Vita4 content and molecular weight did not change much. And finally, we changed the liquid medium composition and we added xylem. And we saw that the Vita4 content remained almost the same, but the molecular weight did increase. We studied the impact of xylem on several different parameters of the system. And we saw that, first of all, the cells were growing more in the presence of xylem. And we also measured a higher peroxidase activity in the treatments with xylem provision. We used these two observations in order to explain why we saw a higher ECL yield in the systems that contained xylem. And as for the ECL structure, we saw that the Vita4 content and molecular weight of our lignin varying depending on the liquid medium composition, xylem provision, and xylem concentration, although in most of the treatments, they were on similar levels. Interestingly, we saw that the concentration of phenyl glycoside LCC was increasing when xylem was added into the lignin polymerization environment. The results of this study can be summarized in the figure that you can see here. So this figure describes the interactions that we think xylem has with lignin. And on the left, you can see that xylem can physically interact with lignin, and maybe by steric hindrance, it can locally concentrate monolignals, facilitating radical coupling. LCC formation is not favored here because ECL is surrounded by water, the addition of which is more favored to ECL than compared to a hydroxyl group from xylem that would lead to LCC formation. However, on the right side, we can see that as we move closer to the surface of the cell, xylem can exclude water, leading to local changes of pH and hydrophobicity, both of which can favor LCC and ECL formation. To summarize these two projects, we were able with this system to characterize lignin without the need of extraction, and also to study the impact of a secondary cell wall hemicellulose on the structure and biosynthesis of extracellular lignin. So moving on to systematically characterize study the impact of extraction on the structure of lignin, we employed spruce and birch wood chips. In softwood species, such as spruce, lignin comprises G units. And in hardwood species, such as birch, lignin contains both S and G units. This difference in the monolignal composition leads to the formation of a lignin molecule polymer that has different structure. So we thought that the impact of extraction would differ as well. So to start our extraction protocol, we tested the impact of a popular mechanical pretreatment, and that is ball milling. So we milled our samples for different milling intervals and for under different milling atmospheres. And our first extraction step was a warm water extraction, with which we removed most of the hemicelluloses, opening up the structure for the next steps. And also we extracted a small lignin population that we characterized. After that, we did an alkaline extraction, with which we got our first lignin-rich extract. And then we did a swelling using an ionic liquid, facilitating further extraction of the residual lignin with a solution of ethanol that contained hydrochloric acid. In the case of birds, we tried two modifications to the protocol you see on the left, and that is swelling directly after warm water extraction, and also removal of hydrochloric acid from the ethanol solution in the final extraction step. In this table, you can see some of the properties of lignin that was extracted from spruce and birch. And in general, we can see that in all the extraction steps, birch lignin has a higher β4 content compared to spruce lignin. And this is because this was expected, because birch lignin is S-rich. And the general trend for both species was that we saw an increasing β4 content as we were moving on with our extraction protocol. Even though we used quite mild conditions in our extraction protocol, we were able to get up to 85% of our lignin in our spruce samples and up to 74% in our birch samples. With pyrolysis GCMS on the birch extraction extracts and extraction residues, we saw a preferential S-unit extraction in the warm water extract, a preferential G-unit extraction in the ionic liquid and ethanol extract, and no specific preference to a unit for the alkaline extraction. We also saw an accumulation of oxidized moieties in the extraction, in the final extraction residue, which is important to point out here because it means that even with the mild conditions that we were using, we can have side reactions that can alter the structure of native lignin. With this protocol, we were also able to isolate some lignin carbohydrate complexes, both of ether and ester type. And an important difference between the two species is that we saw a benzyl ester LCC in the spruce warm water extract and in the final ionic liquid and ethanol extracted fraction in birch, which came after an alkaline extraction. In order to see how these fractions can be used in a popular application, we use these fractions that you see here in order to make lignin nanoparticles. And we did that with a well-established method of solvent exchange. What we saw was that depending on the properties of the fractions that we used, our nanoparticles exhibited different properties as well, size and charge, and also different morphologies. So for example, we had hollow spheres, compact spheres, and snowman morphologies. And we saw that these depended not only on the most commonly studied hydroxyl content, molecular weight, and concentration of lignin, but also on the hemicellulose content of the fraction used, the β4 content, and the S/G ratio. And interestingly, we didn't see one parameter unequivocally driving the formation mechanism, but it was rather a combination of parameters. I'm also happy to have been included in two more projects for which I did lignin analysis, and for both of them, wood aerogels were formed. So for the formation of the aerogels, that was achieved by partial dissolution of cell wall components. And these components diffused into the lumen. Then with regeneration in water, we had the formation of a nanofibrillar network inside the lumen, which was instrumental for the properties of the final material. The partial dissolution of cell wall components was achieved either by the use of ionic liquid and DMSO, or by cold sodium hydroxide. And due to this dissolution, there was an unavoidable mass loss. However, polysaccharides and lignin were lost in a similar way, so we ended up with gels that had a similar composition to the starting material. Due to this formation of the nanofibrillar network inside the until then empty lumen, we have an increased surface area, and that was very important for the properties of aerogels used in hydrovoltaic energy harvesting applications, where we utilize the energy that is produced from the spontaneous evaporation of water. What we also saw that was important for the properties of this aerogel was that lignin was slightly modified, which means that we saw a small reduction in the β4 content. That, however, led to an increase in the hydroxyl content, which means that we had a higher surface charge, improving the properties of the final material. As I said before, there was some mass loss during this process of dissolution. However, not all of the lignin was removed, and it was rather redistributed in the cell wall. And that was very important for the mechanical properties of aerogels that exhibited shape memory properties. And the mechanical properties of this material was in the range of native wood, which is actually quite impressive for an aerogel. These applications are important because they show that severe lignin modification or even complete removal of lignin is not always necessary in order to get useful materials with competitive properties. To conclude, we started by showing that lignin biosynthesis and the structure of extracellular lignin are affected by many different parameters, one of which is the presence of xylan. And then we show that the extraction conditions impact the structure of lignin and the interactions with polysaccharides, such as in LCCs. And also we showed that lightly modified lignin has a high potential to be used in interesting applications, such as lignin nanoparticles, and can add to the properties of wood aerogels. In the future, it would be very important to identify all the biosynthetic pathways that are affected by the presence of xylan, and also an investigation of the extraction impact on other industrially interesting biomasses, such as, for example, energy crops would be interesting. And finally, it would be very important to do an in-depth study of the lignin nanoparticle formation mechanism to find exactly which parameters are driving this process. It is important to remember that for sustainable development to be possible, it's not enough to post hoc find ways to valorize the waste of the industry, but we need to consciously utilize all that nature offers. Before I finish, I would like to take a few moments and acknowledge and thank the Knut and Alice Wallenberg Foundation and the Wallenberg Wood Science Center for funding my studies and research these past 4 years. And also my supervisor, Laura McKee, and my co-supervisor, Francisco Laplana, our lab group in the Division of Glycoscience, and all my co-authors and friends and colleagues at the BWSC Academy, FPT, and chemistry departments, and NGI, NPIS, Lundström, and Håkansson Foundations for RNA sequencing, data analysis, and funding of these experiments. And of course, thank you all for your attention.